Sister chromatids are held together by something called sister chromatid cohesion. And what this does is hold duplicated chromosomes together from the time of DNA replication until they are separated during cell division. So when we're looking at a chromosome like this, it's actually made up of two sister chromatids, which are identical copies of each other connected by their centromeres. And because they are tightly packed, coiled, or condensed, we call them duplicated chromosomes. So anytime that we see two condensed sister chromatids that are attached, we call that a duplicated chromosome. Now here's something that people often miss. Each sister chromatid has its own centromere. We usually only see one centromere spot in diagrams, but there are actually two centromeres, one per chromatid. At each centromere, there's the kinetochore, and that's the structure that the spindle fibers are going to grab onto later when that cell is ready to pull the chromatids apart. So duplicated chromosomes, they're also going to have two arms, and each arm is made up of two parts. A short part called the P arm and a long part called the Q arm. Just think about your own arms. You have a forearm and an upper arm, but that doesn't mean that you have four arms. And this is the same concept here. Each chromosome arm has two parts, but you still only have two arms per chromosome. Now let's talk about how these sister chromatids stay stuck together. We have two main protein complexes involved. First, we have cohesins. These proteins are like molecular handcuffs. They're going to physically wrap around the sister chromatids and hold them together from the top to bottom, the entire length, including at the centromere. And then we have Shugoshin 1, or SGO1. This protein, it hangs out specifically at the centromere. Its name literally means guardian spirit in Japanese, and that's exactly what it does. It protects the cohesins at the centromeres so that they don't get removed too early. So as a recap, cohesins, they are the handcuffs that are going to hold chromatids together. SCO1 is the bodyguard at the centromere that protects those handcuffs. But what actually makes the chromatids separate? That's where an enzyme called separase comes in. When the cell is ready to divide, separase is going to cleave the cohesins, but only along the arms of the chromatids. It doesn't touch the cohesins at the centromere right away, and that's because SCO1 is still protecting them. This really careful timing is going to make sure that chromatids don't split too soon, which could mess up the entire chromosome count in the daughter cells. So if you want a part 2 where we're going to break down how SCO1 eventually gets removed and how separase knows when to activate, comment below. And don't forget to like and follow for more daily science. See you next time.